Hi everyone, my name is Margu Mahyuddin. I am a senior bioinformatics scientist at Bina. I generally work on assessing and improving the accuracy of secondary analysis. More specifically, we have focused on detecting large variations called structural variations. And what we have found by looking at the current tools is that they're not very accurate when it comes to calling these large variations. So we developed MetaSV, which uses an ensemble approach to detect structural variations. And the way MetaSV works is it uses the results of multiple structural variant callers using different methods for calling structural variations, and it merges the results in a smart way in order to get the best possible accuracy across different kinds of structural variations and also across different sizes. Structural variations are large variations in the genome, and the problem is that with the current NGS technology, which uses short reads, it's difficult to detect these long variations, especially if it's insertions or translocations or duplications. In fact, there are a lot of tools which exist to detect these variations, and these tools use a combination of different techniques, and by combination I mean one or more of the following techniques. Like read pair analysis looks for variations in the paired alignment, or split read, which looks for the evidence of SV by splitting the read and aligning it against the genome. Junction mapping, which uses databases of known variations and checks for evidence for those variations. Or read depth, which looks at variations in coverage. And finally assembly, which assembles the read together to construct the SV sequence. And in fact, for these methods and the different tools out, their accuracy varies by size of the SV and also by the kind of SV. So there really is no one solution which fits all. Each tool and method has their strengths and weaknesses when it comes to specific sizes or kinds of SVs. So this slide just shows a summary of the different SV detection methods which I talked in the previous slide, and it's a more visual representation of the kind of evidence is used for figuring out whether a particular SV exists in the genome or not. So because each method and tool has their strengths and weaknesses, we have developed MetaSV, which integrates across multiple methods and tools to get a better quality set of structural variations. And in fact, there has been prior work in this domain, like SV merge or ISVP, but each have their own limitations. So MetaSV actually tries to be more comprehensive by detecting more kinds of variations, and also tries to integrate the results from different tools and methods in a smarter way. So the reason integration is important is that prior work has shown that if multiple algorithms call the same variant, then it's most likely a higher quality variant in the sense that it's more accurate. In fact, if we see that from the 1000 Genomes project, if 3 algorithms were able to call a particular variant, that variant actually had a very low false positive rate. So in MetaSV, we adopt this approach and we look for evidence of the same SV across different methods. If multiple methods call the same SV, we report it as a high quality variant. Now that we have established that integration is useful, we also need to integrate them in a smart way. If we simply merge the results from all the different tools and methods, we might end up having a very, very high false positive rate. And therefore, we have developed MetaSV to be method aware. So it uses the strengths and weaknesses of different tools. For example, some tools are more accurate, like Breakdancer can only report variations within a couple of standard deviations of the insert size distribution, while Brexy can report variations within one breakpoint accuracy. So we have to be aware of these strengths while we'll try to integrate across different tools. And in addition, we have realized that the existing methods, they don't really do a good job at calling long insertions. Therefore, we have also added an enhancement that uses a soft clip based method to detect long insertions. And this soft clip based method in combination with assembly can yield a very high accuracy for calling insertions. So once we have detected and merged the SVs from different tools, we do assembly and followed by alignment against the reference genome to get as precise breakpoints as possible. In order to assess the accuracy of MetaSV and also study the impact in terms of improving the accuracy over the existing tools, we used both simulation and real data. For simulation, we used the VARSIM framework developed at Bina to generate NGS reads at high coverage of 50x with the Illumina Platinum Genome's error profile. And we also used a high reciprocal overlap of 90% with a wiggle of 100 base pair to make sure that we only capture high quality and more precise calls. So this is a more restrictive kind of assessment. And we see that the performance for different tools varies across different sizes. And in fact, the MetaSV, which is the top blue line, which achieves the most stable accuracy. In fact, it is the best performing for each size bin. In addition, the performance of MetaSV doesn't drop much except for the smallest size, where because each tool which is used to as input to MetaSV, because they don't perform very well for those sizes, therefore MetaSV performance drop for those sizes. For real data, we actually use the Platinum Genomes project data for NA12878, and we compared the detected SVs, specifically deletions, against the high quality deletions from complete genomics. So for real data, we used the NA12878 sample from the Platinum Genomes project, which is again like roughly 50x coverage for whole genome. And we compared the MetaSV results against the high quality deletions from complete genomics for NA12878, and we achieve more than 90% sensitivity. Since complete genomics is a different technology compared to Illumina, this 90% sensitivity also implies a high level of accuracy for MetaSV. In conclusion, MetaSV was able to improve the accuracy for SV calling by integrating across different tools and methods in a smart way, and as a result achieves the most stable and the best accuracy for calling SVs.